Most of you know me, my name's Dr. Hayes, I'm the head of chemistry here, so I'm just here to introduce Dr. Ben Pilgrim, so he's going to come and talk to you about chemistry beyond the A-level and the Chemistry Olympiad. He's been to quite a few establishments so far, started his career in Oxford, moved to Cambridge, and has recently just started his research group at the University of Nottingham, which is where I went, and a fantastic place that you should definitely have on your UCAS hit list. And he's come to talk to you about all the fun things that we do above and beyond the A-level. So we've definitely had some fun with these at King's. I hope you have at your schools as well, but yeah, they're not easy, and he's to blame.
Hello everyone, well thank you for having me today. So yes, I'm Ben Pilgrim. I have a research group at the University of Nottingham, been there about 4 years now. But I also run the Chemistry Olympiad competition in the UK. Who's, just a show of hands, who's heard of the Chemistry Olympiad? Who's had a go at it already, maybe like previous year? Okay, who's going to have a go at it this year? That should be everyone. Okay, if you don't know what it is, I will explain on the next slide. So it's probably actually the biggest single chemistry event or program in the whole world. So it's I'm going to stand here, so you can see, and not fall off. Competition to find the world's best pre-university chemistry students. About 90 countries each send their 4 best students to an international final, which happens in a different country each year. And at that international final, students win gold, silver or bronze medals for their country, so a bit like the Olympics. But for chemistry, here, so Ryan and Sasha are winning gold medals a few years ago. And so with each country then runs a national round to select their team. So that's what we do in the UK. This was last year in Switzerland, so there's me and several of the other mentors at the back, the 4 team members at the front, and they came away with 1 gold and 3 silver medals last year. Here's their team mascot, Paddington Bear. So that was in Switzerland last year. Didn't happen in person over COVID. A few years previously in France, so very stereotypical France picture there. They're giving the team mascot a baguette there, so again, very stereotypical. And again, they won, I think, 1 gold, 2 silvers and a bronze that year. So it kind of culminates in this international final, and the UK often does pretty well against students from all over the world, and it's a fantastic kind of cultural experience, meeting so many other people that are as enthusiastic about chemistry as you are. So how does it work in the UK? So there are 2 rounds, so this was last year, so nearly 12,000 students took the first round, which is a 2-hour theory paper that's sat in schools. Anyone can enter. You just need to get your teacher to register you. So if your school doesn't do it, I know some schools here do it, quite a few you put your hand up, but if you haven't heard of it at your school, a teacher can sign it up. It's free, so it doesn't cost the school anything. It'll be in January, and you just need to find, a time. Usually they do it in 3 periods, or you get out of lessons for a couple of hours in the afternoon to sit the paper. And the paper is tough, but it's quite good fun. I'll talk through some of the questions in a moment. And then the very best of those students kind of go forward in the competition to the second round, which takes place at the University of Nottingham now. And we have some more theory and practical tests for students, and some training and teaching you some new things. And eventually we pick the 4 students for the team. So it's tough. I think that was mentioned in the introduction, and I often write some of the toughest questions. So it is a hard exam, but there's a lot of scope for finding out new chemistry. You don't actually have to score that many marks in order to get a kind of gold, silver or bronze award in the national round, and they're very, very useful things to put on UCAS applications for university, because again it shows that you're interested, you've got kind of commitment to the subject beyond just the A-level course, and if you do get one of these kind of higher awards, it does show that you're very good. Say the Olympiad paper is significantly more difficult than an A-level exam. So this is kind of a mark distribution from last year. So this looks very different to how the mark distribution on an A-level paper would look. So on an A-level paper, I don't know what percentage of sort of raw marks you might need typically to get the top grade. It'll vary year to year, but it doesn't usually have a mark distribution that looks too much like this. So actually the kind of median mark, is quite low, and the mode, is a distribution that's really sort of skewed in this way. But there are, not that many people actually, get zero, right? So a good chunk of people, can approach some of the questions, but there are really, really tough bits as well that allow us to separate out the real top students who then get to go through to the next round. So what sort of thing do we ask about? So we try and pick things to show the relevance of chemistry to society. So A-level, understandably, fixes on certain topics. You've probably done the Harbour process, for example, to make ammonia. There's certain things that form the part of the A-level syllabus, and there's not time in the school curriculum to teach all the different ways that chemistry can affect things in terms of products, in terms of combating climate change and environmental issues, drugs, new smart devices, all sorts of things. So we try to take our inspiration from stories that have been popular in the news in the past year, or other things that we think people might find interesting. So when we were writing last year, it was going to be in Switzerland, and so we were thinking a question on cheese. And at the time, when we were writing this last kind of summer, seems a long time ago, Liz Truss wasn't actually yet Prime Minister, but I don't know whether any of you, hopefully this video works.
I want to see us eating more British food here in Britain. At the moment, we import 2/3 of all of our apples. We import 9/10 of all of our pears. We import 2/3 of our cheese. That is a disgrace.
So a bit of a strange video there. It seems a little bit of a while ago. When we first started writing this question, she was yet to be Prime Minister, but at the time students sat the paper, she'd already stopped being Prime Minister. So you had to go through quite a few drafts. But with the Olympiad in Switzerland, we tried to do a question showing how kind of chemistry is important in the cheese making process. I had good fun making a little cartoon for the question. And so it kind of started off with thinking about, cheese comes from all over the world, right? But the fundamentals of the cheese making process are actually the same at a kind of chemistry level. So we have lactose is the sugar in milk, and that gets fermented to lactic acid. And so the question starts off with some stuff that hopefully is not too challenging if you're kind of doing chemistry A-level. So it's talking about the acidity of this carboxylic acid proton in lactic acid. So asking to draw the conjugate base, so that's just to draw the carboxylate, to calculate the pKa. So again, that's an equation that you should be familiar with A-level. And then talking about how, for example, lactic acid is more acidic than propanoic acid, and that's because we have a hydrogen bond between the OH here and then the deprotonated form here, and that makes it more acidic. So there's sort of some straightforward bits to kind of get going on this topic. Now I'm not going to go through kind of the whole question here. You can find all of these online, and I'll show a link to them at the end. But Swiss cheese is famous for its holes. Okay, I'm sure you've all had some sort of holy cheese before. So does anyone know what makes the holes?
Gas.
A gas, yeah. Which gas do we think?
Carbon dioxide.
Carbon dioxide, yeah. So that's because there's another bacteria that's in the cheese fermentation process that breaks the lactic acid down into carbon dioxide. And so this question went on to then think about how we make these bubbles within cheese. So if you're a cheese manufacturer and you want the right number and amount of bubbles, then you've got to control these variables. And so we can first think about maybe a typical size we might have of a bubble, and then we're thinking about its volume and using the ideal gas law, thinking about how much carbon dioxide we would then need in that bubble. But it's actually slightly more complicated than that because it's an equilibrium problem in that we can have the gas as a gas bubble, but we can also dissolve gas in liquid. And although we don't usually think of cheese as being liquid, the cheese sort of solidifies, over the cheese manufacturing process. It solidifies from the milk. So you have to consider an equilibrium between the gas carbon dioxide in the bubble and carbon dioxide dissolved in the liquid, or whatever we're going to say the state of the cheese is. And you have to consider that equilibrium to work out how big these bubbles grow. So that's what we looked at in this question. So going on to another one that we picked. So who has seen E5 and E10 numbers on like petrol filling stations? A few of you. So there's two numbers that we typically get on a fuel. Okay, so one is a number that will be something like 95, 98. That's the octane number. They're kind of telling you essentially how well that mixture combusts. Higher leads to better combustion. And then there's an E number. It was E5. And about a couple of years ago, the UK switched petrol fuels over to E10. Now the E here stands for ethanol, and it means that this fuel can contain up to 10% ethanol. Now, why is that important? Well, there were some fuel shortages a couple of years ago, and this switchover was thought of as one of the reasons contributing to this, but it also is very important from an environmental point of view. So again, we kind of talk about both of these numbers in the question. So this compound here, 2,2,4-trimethyl pentane, that's one of the standard compounds in petrol. It's highly branched isomer of octane, and that's good for that octane number, keeping it high to avoid the knocking in the engine. Now, adding ethanol also helps to reduce knocking, but as I say, the main purpose of adding the ethanol is that ethanol is made from sugar, and if we think about, we've grown our plant, essentially we've made glucose, we need to convert the glucose into ethanol, and then eventually when that ethanol is burnt, all of that carbon dioxide that comes from that ethanol was removed from the air by growing the sugar plant to begin with. So in terms of kind of net contribution of carbon dioxide to the atmosphere, it is better because we've taken it out and we've grown the sugar, made the ethanol, burnt it and it's gone back, rather than releasing carbon dioxide from an oil deposit that's been locked out of the carbon cycle for millions of years. So this question was asking about some of the kind of chemical equations in the reactions of glucose, and then finally kind of building towards a calculation looking at kind of the lowering of CO2 based on swapping from E5 fuel to E10 fuel. And it's a good few percent saving in terms of the amount of CO2 that's emitted per mile that you drive. And then, of course, if you scale that up to the number of miles that are driven every day, it's a significant effect on reducing CO2. Okay, so I presume everyone's done a COVID test at some point in their life. You may have wondered how they work. So this is a kind of simple cartoon. You're kind of, you're swabbing your mouth, your throat, you're kind of dabbing it on here, and then your solution is flowing along the test, and of course you don't want to get your kind of dreaded double line, or maybe you do and you get a day off school or whatever, but kind of how does this work? So it uses kind of antibodies essentially that bind very tightly to the spike proteins and so on the virus. And it's essentially an equilibrium problem in chemistry. So in order to design this test, it's kind of building on that stuff you learn at school. So you learn about equilibrium constants at school. The actual amount of virus particle is very low, and that means we generally need something that binds very tightly, has a very high equilibrium constant for binding, so that we can then have a detection with a relatively small amount of the sample. So this is kind of based on some sort of typical figures. So here, about 7 times 10 to the 6 virus particles if you're a positive test and you've swabbed yourself and you've got your little sample. And then let's say that each virus has 20 spike proteins, and these are the things that our antibody is going to bind to. So we can work out the concentration of that, and we need concentrations to do our equilibrium constant calculations. So the sample comes through here, and then in the conjugate pad, it's going to pick up these nanoparticles, so they're gold nanoparticles. Now, the colour on most COVID tests is red, right? And it's quite strange to think that the red colour is actually coming from gold. Why is it not gold colour? But when things are very small, in the kind of nanometer length scale, then their colour is controlled by, let's really say, more complicated things. And they can appear different colours. So nanoparticulate gold often appears red. So that's, and when that then gets deposited on these 2 lines, then that's telling you that you've got a positive test. So there's quite a high concentration of these nanoparticles kind of on this conjugate pad, and we can consider these different equilibria. So here the spike protein is binding onto the nanoparticle, and it's a reversible binding, but it's got quite a high equilibrium constant. So it's a strong binding, so we can detect a relatively low number of virus particles in total. And again, I won't kind of go through, you can look at all these later, but this question built through kind of looking at, the equilibria that had to happen in order for first the virus to bind to the nanoparticles, and then for the nanoparticles to bind onto the test line in order to give you the positive test result. And eventually kind of working out, the minimum number of virus particles you would have to give you a positive test. Sticking with COVID, the question we had in a different year is this molecule hydroxychloroquine. So this was a favourite of Donald Trump. So he suggested a number of things to treat COVID that just wouldn't have worked or just completely stupid. So for example, shining UV light inside the body, injecting disinfectants, and in particular he said he was taking this hydroxychloroquine that was meant to prevent him from catching COVID, and then a few months later he caught COVID. So this doesn't work, but nevertheless it is a drug, and it's used for other things, and people synthesize it and so on. I usually design the cartoons here, so this is quite great fun with designing the cartoon for this one. And again, this is where, those who say kind of the Chemistry Olympiad is always hard. I'd hope that some of the starting questions, at least here, were relatively more straightforward. So this, starting with benzene, this is one of the standard A-level chemistry reactions you would learn that happens on benzene. So we are nitrating benzene to start with, and then we're chlorinating it in the next step. And if we look here, for example, we can work backwards and get some information, okay, from the thing that we're going to about, for example, where the position of the nitro group and the chloro group are relative to it. They end up meta to each other. This is then reducing the nitro group to an NH2, and the synthesis goes on. But just want to say that whilst there are some more difficult bits, there are some hopefully more accessible bits in each question as well. So again, the importance of chemistry in kind of the drug industry. So this was another fun article that was out a few years ago. So the molecule phosphine was detected on Venus, and on Earth, phosphine is only really produced from living things, and so its presence on Venus maybe indicated that there were living things on Venus. This has, I think, since been kind of, not many people necessarily agree with that this was true, I guess now. There's been a lot of debate about this in the sort of scientific literature. But we decided, to use this as inspiration for a question, so asking people, to draw the shape of phosphine, and the fact that we don't have phosphine in the Earth's atmosphere is because it would react with oxygen, so the equations for that, and then some other equations here talking about various ways or reactions that might produce phosphine. But that would require, for example, these chemicals to also be present, on the Venus surface or whatever. Anyway, so that was drawing on a more recent news story. And then finally, one on sun cream. So sun cream is quite toxic in some ways to certain sorts of marine organisms, and some islands are actually banning certain types of sun cream because it kind of goes in the water when people swim in the beaches. And so this one here, based on this quite simple molecule, was banned a few years ago. And again, the synthesis of this, here you have a product, so you have to work backwards. You've got some formulae here. Aluminium trichloride, you might recognize as one of the reagents that you need in a Friedel-Crafts type reaction of a benzene ring. And so you might then be able to think about, okay, well, where are we breaking the bond, going back, and so on. So they're just a kind of a snapshot of some of the areas where I hope kind of the Olympiad shows how chemistry is, really important to all sorts of areas of kind of modern life and modern society. And if you want to kind of find out more, there's over 20 years of past papers on the website of the Royal Society of Chemistry. And if you want to, you can find it very easily by Googling. I guess that QR code might not work with that pixel thing out there, but certainly go and have a look at it on there at some point. So, where do I come into this? So, I have, the RSE very nicely gave me a prize for this last year, but I've been kind of running the Olympiad for about 10 to 15 years now, so I've been involved in it for a long time. But what do I do kind of outside the Olympiad? Now, I don't work on coronavirus, which is here, but I do work on something that we refer to as sort of self-assembled systems. So, do we know roughly how big this coronavirus particle is? Someone says nanometers. So, anybody else got any guesses? I mean, nanometers, some amount of nanometers is correct. So it's kind of a bit over 100 nanometers in size, okay? But this is a structure that's made by self-assembly. So the proteins, the lipids that make up the grey kind of shell, they all kind of reversibly come together and stick together to give the structure of the virus. In fact, that's true for all sorts of viruses. This is the poliovirus, so that's a bit smaller. That has kind of, its entire coat is made up of protein rather than a mixture of protein and a fat. DNA, that's is very long, but across the width of the DNA, it's only a couple of nanometers. But that is a very specific self-assembled structure, in that the two strands will pair up exactly with their complementary strand due to that sequence of hydrogen bonding. But again, that's a kind of another thing in this broad category of processes that I would say we would refer to as self-assembly. Okay, so things have kind of, the design principles are encoded in the pieces so that when they're in the presence of each other, they can undergo reversible interactions, they come together, they can fall apart, but they end up assembling to some kind of structure because that's the most stable state of the system. Now, this is a termite mound. This is much bigger, okay, this is a few meters in size, but there are some principles of self-assembly here as well. There's no single kind of architect termite that drew up a plan for this mound on kind of day one, okay? All the termites know that, okay, if I'm assigned my role as a builder, then I'm going to go and stick an extra bit on the outside, but I don't want to block off a tunnel. So there's certain rules that they will follow when building a mound, but it's not like someone's sort of sketched out the structure from scratch. So there are certain principles of self-assembly that apply in these much bigger systems. This is the Great Barrier Reef. This is hundreds of kilometers in size, but again, some of the principles of self-assembly apply to this as well. So, gonna ask for a couple of volunteers to come up. Okay, you two there, if you put your hands up to begin with. So, what we have here is we have some magnetic blocks, okay? And you put them in here, you shake them up, and they're gonna assemble into a dodecahedral structure there. So I'll give both views. So before you start, so you need to, don't do it yet, you need to shape them in order to make them kind of stick together. But we need to shape them with, I would say, kind of medium strength intensity. If you shape them too slowly or too weakly, you won't give them enough energy to move around and kind of start making the bond. But if you bash them about too vigorously, you'll kind of bash apart some of the ones you've already made before they can grow into the whole structure. So we'll say like 3, 2, 1, go. So shape them up. Well done over here.
Can't do that. I'm seeking a bit more intensity. One part, listen. Shake it.
Okay, well, there we go. Thank you. So this is kind of showing us the self-assembly process. So what makes this successful? We need a kind of reversibility in the interaction. So these are hydrogen bonds between water molecules. We get hydrogen bonds between the DNA strands as well. They're a type of interaction we might use in this sort of assembly because they're of that kind of right kind of strength, that kind of Goldilocks, not too strong, not too weak strength, so that the bonds between the ones we want are strong enough to hold together, but if something comes together in the wrong way, it's not going to be stuck together permanently. We also make use of symmetry, so this is a quite highly symmetrical structure, this is a dodecahedron, and symmetry means that you can use the same piece multiple times to make a far more complex structure far more easily. The structure of those viruses had a lot of bits repeating, and again that means that there's a lot less design material, a lot less kind of genetic code is needed to code for all the bits of the virus if you can use the same bit multiple times. So symmetry is very important as well. And kind of cooperativity, this is where sort of correct interactions reinforce each other to help bias the system to kind of only make the ones you want. Now, in the video here, I had red and green in the same bottle, and they selectively paired up with each other. And, well, I won't tell you the answer to that now, but maybe you can think on that and see if anyone knows at the end. How can red and green have selectively found each other? So what do I do in my research? So I make these self-assembled structures as well. So we use reversible bond interactions. This is the reversible interaction, for example, between an amine group and an aldehyde to make an imine. And then these two nitrogens can coordinate to a metal ion, shown as a kind of 2+ metal ion here. Now, if we have this particular molecule here and two of these, we can kind of do this reversible imine formation twice. And we can coordinate to a metal here and a metal here. And this actually allows us to make a kind of edge piece to assemble into a tetrahedral structure. So this is a kind of visualization of what this structure might look like. So we call these things cages, metal organic cages. And they can be made just from mixing a few simple pieces. They undergo this reversible interactions and assemble into a structure such as this. And well, why are we interested in these structures? So we're looking at binding things inside for catalysis. So a bit like an enzyme has an active site in the middle of it to do reactions. We have a three-dimensional architecture here, and we can do catalytic reactions on the interior of these cages. The metals involved in some of our cages are, for example, what we would call paramagnetic. They have unpaired electrons, and that can be very useful for techniques such as MRI, which is the same phenomenon as an NMR spectrum, but just in three-dimensional space, so it allows you to take an image of the body. And the cavity inside some of these cages could potentially be used to bind a drug, so there's applications in drug delivery. We're looking at cages that can change size or shape in response to certain stimuli, for stimuli-responsive materials. And then we're also looking, that video doesn't seem to play, but this is an example of something that we call an interlocked molecule. This is a ring locked on an axle. And the reason we're interested in these is it allows us to build molecular machines, which is another way of kind of making molecules do quite complex tasks, but probably don't have time to really go into that today. So I'll leave it there, and thank you for your attention. Happy to take any questions. Yes?
Is there a reason why you chose Nottingham?
Is there a reason why I chose Nottingham?
Yeah.
So I guess kind of when you are looking for a job in a university, not all the universities will necessarily be advertising for a job at the same time. So it's unlike kind of when you apply for undergraduate that the university might be taking on 150 people a year. In terms of members of staff, a university might only hire once every 3 or 4 years. So a lot of it was just down to sort of who was advertising, in the year that I was looking for jobs and so on. But it's a very nice place. So if you haven't been there, the campus is a very lovely open green campus, but it's also close to a major city. So I'd say kind of the university has the best bits of both a kind of campus style university, but also a city university as well. Hello.
Do you have any jokes you thought of for this year's Olympiad?
Do I have any jokes? I have thought of a couple of them, but I can't tell you what they are at the moment because that would be giving away some of the questions. But so we often go for things that have been in the news. So if you think of the sort of the stories that have been in the news in the past year, then that might give you some ideas.
Is a tetrahedral shape more beneficial over other shapes to bind to like catalysis or the drug receptors, or is it any shape that works?
So there's nothing, I guess, inherently magical about a tetrahedral shape. So a tetrahedron is one of 5 platonic solids. I don't know whether you know what that means. Some of you might have heard that they're sort of mathematical shapes. So a cube is one as well, an octahedron is one. this dodecahedron is one. So there are 5 shapes that have all the faces, the edges, the corners the same. And that means that if you make them up from kind of a face capping piece, as we did there, a pentagon, or a kind of edge capping piece, as I was doing in the molecular example, you can use only one type of piece. So that's kind of why high symmetry shapes are good. We've made things such as cubes and stuff as well. When it comes to kind of making them with chemistry rather than making them with 3D printed models, you have to think about, do the coordination numbers match, for example. So at the corner of a tetrahedron, we had an octahedral metal which wants to bind 6 things, and we had 3 ligands meeting at that corner, each of which can make 2 bonds to the metal. So that all added up. But if we had tried to make an octahedron, for example, then we'd have 4 things meeting at a corner, and that would be a different kind of thing that we'd need to work out. So some shapes are easier to make than others.
Can you give an example of one of those shapes that get changed by a stimulus? What kind of stimulus would be useful to have it change shapes?
So we can do chemical reactions. So it can either be a chemical that you're trying to detect that leads to a change in shape, and then sometimes that change in shape leads to a change in colour or a change in solubility. So there's some way of reading out, detecting it. Light, we often add sort of photo-responsive chemical groups in there that can sort of isomerize, a bit like how vision works in the eye with a kind of cis to trans isomerization that can change shape quite significantly. You can also get some sorts of molecules that, as you raise or lower the temperature, they kind of curl and uncurl, more like polymeric molecules. They curl and uncurl, and that can change solubility.
What sort of applications would we see from chemical machines, like through the ones that you gave an example for?
Yes, so all of complex tasks in biology are done by molecular machines. So for those of you who don't know what molecular machines are, they're things such as pumps, so they can control differences in concentration, for example, inside and outside of a cell, by actively pumping, for example, protons or ions one way or other through a cell membrane. You get things such as walkers that will kind of hold a little vesicle and will kind of move it across a cell. You can get motors. So these are things that, for example, propel bacteria. They have like a kind of rotary kind of motor on their back. So designing kind of synthetic equivalents of those, I guess, allows us to make far more complex molecules. Because the molecules we can make synthetically, we can do lots of cool stuff with, but we're a long way away from the sort of sophistication that's evolved in biology over billions of years. So that's why we're trying to, develop more complex molecules that enable us to have a go at some more complex tasks. I don't know how much time I've got left. Is someone? Well, keep going for the
Are
moment.
you supposed to have an A-level certificate to actually take part in the Olympiad?
No, so you don't have to be doing A levels. You can be doing Scottish Highers or IB or things like that. And you don't have to have already done A levels. You can take the Olympiad. You can take it in year 13, year 12, year 11 if you are really keen. Where else do we have? Yes, sir.
In the design of your chemistry compounds, do you take inspiration from the biological systems?
Yeah, so if you don't want to have to design something from scratch if it's sort of been designed already. So yeah, you can look at biological systems for things that bind or things that orientate in particular ways and think, okay, how can we adapt that principle in our system? So yeah, we do look at them for inspiration, both in terms of structure and the exact chemicals we use, but as I was sort of saying in answer to a previous question, in terms of function, we look at the functions that biology has got in its complex molecular sort of machinery, and can we then start to design those functions into artificial systems. Yes?
So, you showed the example where if you put both of the shapes into one bottle and it self-assembles as separate, is it just the orientation of magnets and the distance between each edge?
So it is to do with the orientation. The distance is the same, and maybe I can show people now. The distance is the same. So magnets have a north and a south pole.
It's sort of like chirality.
It is chirality, yeah. So magnets have a north and a south pole, and obviously the bottom edge, I don't know how clearly you can see this, the bottom edge is thicker than the top edge. So kind of if we put the magnet down on the table, we kind of look at which pole of the magnet is facing out. This one goes sort of north-south, north-south, and the other one goes sort of south-north, south-north. Now what that means is two of the same edge can stick together like that, but two of a different type of edge, they can't stick together like that because the magnets are the wrong way around. So two of a different sort of edge can only stick together like that. Now if you stick them together like that, there is no way for a third one to come in and cooperatively form interactions with both sides. But if you have two of the same, then the third one can come in and therefore make two sets of interactions, and then this piece is now much more strongly held together than just a kind of dimer, if you like. So yeah, it's to do with the fact that the arrangements of magnets in the structures is chiral. They're kind of enantiomers of each other.
Yeah, last question, that was it. Thank you. So thank you. Could everyone give a round of applause?